to everybody. Welcome to the 32nd seminar of the Hellenic Initiative Against Alzheimer's Disease Seminar Series Bridges and the 15th satellite event of Genedis 2026. It is a great pleasure to have today as speaker Professor Leonidas Stefanis, who will talk about genetic synuceinopathies from the bedside to the bench and back. Professor Stefanis obtained his medical degree and PhD in molecular medicine at the University of Athens, and he trained as a neurologist and neurobiologist at Columbia University in New York. Since 2003, he is back and serves as a clinician and scientist at the Medical School of the University of Athens and as researcher in the Biomedical Research Foundation of the Academy of Athens, where he has established a lab of neurodegenerative diseases. Since 2017, Professor Stefanis is the head of the first department of neurology at the University of Athens at Genedis Hospital. His main clinical focus is movement disorders, and in particular Parkinson's disease and other synuceinopathies. He runs a clinic for movement disorders, offering opportunities for genetic, biomarker, and other translational research. He is following the largest worldwide cohort of subjects with genetic synuceinopathies, and he has established the European Center of Excellence on Rare Neurological Diseases. He is further examining the interrelated pathways of alpha-synuclein regulation and associated neurotoxicity, with an emphasis on the involvement of protein degradation pathways. Professor Stefanis, many thanks for being with us today. The floor is yours.
Yeah, thank you, Professor Alexopoulos, and I want to thank you all for inviting me. It's a great honor. So I'll go right into the subject. Already mentioned that this is work conducted both at Genedis Hospital and at the Biomedical Research Foundation of the Academy of Athens. So this is the outline of the talk, introduction, then our first foray into genetic synuceinopathies through the famous HT3T cohort. Initially studies enabled for a European grant, then the PPMI in the last few years. I wanted to stress asymptomatic carriers of this mutation, and then biomarker studies, something that we've recently embarked upon, the connection between the SNCA pathology and ApoE. A new mutation we've uncovered recently, the A30G SNCA mutation, and some studies in the more biological level of that mutation. And then just finally ending with stressing the importance of genetic cohorts. So for those who are not well-versed in synuceinopathies, these are nervous system disorders where there's abnormal deposition of this otherwise normal neuronal protein alpha-synuclein. It's a very abundant presynaptic protein in the nervous system, but in these conditions, it deposits abnormally in the brains and in the peripheral nervous system, and even in other organs, it seems, of patients with these conditions, Parkinson's disease, dementia of Lewy bodies, and multiple system atrophy. Of course, the panorama of genetic underpinning for Parkinson's is very rich, but SNCA, you see on the top there, was the first gene identified with a definitive genetic link to Parkinson's disease. There's, of course, many, many others since. This is the gene with the introns and exons and the regulatory regions. At the level of the protein, it's a relatively small protein, 140 amino acids. It has an amphipathic region and terminal domain with which it interacts with membranes and other proteins. There's a C-terminal, acidic terminal, and there's a phosphorylation site, which is quite important. We'll go over that at the very end. And then in the middle, there's this NAC domain, which is very important for the aggregation propensity of the protein. And shown here also are a number of point mutations that have been identified in the gene, leading to autosomal dominant forms of the disease. And this is just a genotype-phenotype correlation. of course, this slide is all the slides of the presentation will be available, but I won't go into it in any great detail. But suffice it to say that there's a general pattern, but there's also differences amongst the different families with point mutations identified, and there's even heterogeneity within the same family with the same mutation. But generally, all these are more severe forms of Parkinson's compared to the classic idiopathic form, and you see the mean age of onset is quite early compared to classical Parkinson's. So this is point mutations leading to autosomal dominant disease, and then we have triplications and duplications, so multiplications of the gene locus also leading to Parkinson's disease, autosomal dominant fashion. And here again, there's a genotype-phenotype correlation. What's very important to stress is that triplications are much more severe compared to duplications of the gene locus in terms of penetrance, in terms of age of onset, and severity of the disease once it develops. And I'm not going to this at all, but just to stress that genetic link is not only in the rare familial forms, but also just in sporadic Parkinson's disease. The highest hit on GWAS is actually, you see here, the SNCA locus is on chromosome 4. So SNCA gene is also encoding for alpha-synuclein, is genetically linked also to sporadic disease. And it remains the highest hit after all these years in multiple populations. So how did we get involved into this? We started looking at the families with the original mutation, the 53T mutation, which had been identified in 1997 by Polymeropoulos and colleagues in families in the Peloponnese, essentially, in the area of HIAAAD and surroundings, and also in the famous Contursi Italian kindred. That was the science publication from the Nussbaum lab in 1997. It has been rarely described in individuals of different ethnic background, and generally almost all the cases are Greek or Italian origin. What had been reported before is that it's classic Parkinson's disease dementia, commonly develops rare presentations of dementia of Lewy bodies. Some cases resembled a little bit multiple system atrophy. The age of onset was between 45 and 50, and at the time we started being involved in this, there were about 70 cases reported. So at the time we were setting up this Hellenic Biobank for Parkinson's disease at the Biomedical Research Foundation of the Academy of Athens, together with my colleague Dimitris Vasilatis. And we started some basic demographic clinical information, and initially just with DNA, but over the years we've been doing more biosampling, more biological material that you see here. So we got involved in a European project that was studying Mendelian forms of Parkinson's across Europe, and through that we're able to fund efforts to really go after the families that had been already identified, look at descendants of those families, relatives, but also looking at our outpatient clinic population and see if we could identify in those we are seeing in the clinic any kind of new families with this mutation. And this led to this work around the MEFOPA project, where we actually looked at 111 patients who were not linked to any of the known families, and had a high risk of being a genetic just because they were early onset or had a strong family history. And actually, a sizable percentage of those 5 out of 111 had this mutation. So it's not infrequent in Greece, especially if you have a patient with a autosomal dominant family history. They have an early age of onset. They're from the Peloponnese. It's almost, you don't have to think much about it. They almost certainly have this mutation. And this involved also outreach activities. This is the team going out actually, it's not just HIAAAD, it's also Corinthia. This is actually in the area of Stymphalia. If anyone's been there, it's a beautiful area in mountainous Corinthia. But there are other areas also where there are clusters of these families, and we go visit them. This is the type of families we see. You see autosomal dominant pattern of inheritance hitting almost every generation, high penetrance, but not complete. You see here, this case here, at age 87 is a carrier of the SNCA-AFT3T mutation, and yet she's asymptomatic. And there are other, of course, asymptomatic carriers. So another effort we did at the time was kind of to gather all the cases we could muster, using different approaches, both new families and old families. And we studied 22 patients and 8 asymptomatic carriers. I'm not going to go into the details, but it was the mean age of onset was what was earlier described, around age 45. What was very interesting, these asymptomatic carriers. There was a general pattern that they were quite severe, especially in the progression, and especially regarding non-motor symptoms, psychiatric symptoms, psychosis, dementia were more common as the disease evolves. There was quite a remarkable phenotypic heterogeneity on the other hand. Like I mentioned, some carriers at an advanced stage had not developed the disease, very rare, but it happens. Penetrance is around 80 to 90%. And then we had some cases with a very early age of onset with remarkable brain atrophy and a frontotemporal dementia-like presentation as well, kind of the other end of the spectrum. We've continued this work through our involvement in the PPMI study. This is a worldwide study funded by the Michael J. Fox Foundation. We've entered the study since 2014, and our main contribution in the beginning was just the recruitment of this very rare genetic cohort worldwide. But over the time, we've evolved and we've included both other genetic cases, but also prodromal cases, new onset Parkinson's, and so forth. It's a very detailed protocol as opposed to the MEFOPA protocol, which was quite basic. Here, it's a very detailed testing, imaging, and this is repeated every 6 months to a year. And through this, we're able to recruit more subjects in the study and study them more in depth. And you see an example of this here, where we've done a kind of a more in-depth neuropsychological testing. These are cases, 18 cases compared to 18 idiopathic PD from the same PPMI cohort. And what we're looking at here, the main thing, we're looking for differences, especially in the neuropsychological profile of these patients. These are patients at around 5 years since onset, so around 50 years of age at the time. And what we found is that overall in the MoCA, just a general global assessment of cognitive function was not significantly different, although there was a trend, but there were aspects of neuropsychological testing that were specific, and in particular, frontoparietal network, as exemplified, for example, by the Benton line orientation test, by the letter number sequencing test for frontal lobe functions, but also from visual tests for the parietal visual spatial tests reflecting parietal lobe function. They were significantly more impaired compared to the idiopathic PD. So this was a specific network that seemed to be affected. It correlated also with, which is interesting, with markedly lower olfactory function in the genetic AFT3T cases compared to idiopathic PD. So these we thought were kind of core features of synuceinopathy. More recently, we've been able to do a more prospective study, follow-up of these subjects. At time point 1, you see here, they're about at the 5-year point we mentioned before, and we see them evolving over 3 years, and you see how the MoCA test reflecting global cognitive function deteriorates in blue in our cases compared to the controls. Controls here are actually idiopathic PD. They are not controls. So those remain stable over time, whereas our cases deteriorate. Same thing with UPDRS2, which reflects the global motor function of the patients. It advances considerably much more than in idiopathic PD. And very importantly, also global function, as assessed by the Schwab in England, also deteriorates in our subjects, whereas it's relatively stable in idiopathic PD. So really showing this progressive nature of this disease, and especially regarding non-motor functions and cognitive functions, which may be appropriate endpoints for disease-modifying trials. Imaging also, we did studies on imaging, DAT scan, which reflects nigrostriatal degeneration, again comparing our cases to those that were matched for age, disease duration, and sex, on whether there was excessive nigrostriatal degeneration. Of course, all patients with Parkinson's have abnormal DAT scans, but what we found here is that it was much worse in our cases with a similar disease duration compared to controls. Controls are again idiopathic PD. And you see here examples in the bottom row are cases where you almost don't see at all the uptake of the irritation in the stratum because it's so low. It's called burst stratum. So again, indicating this more severe course of the disease. So overall, we've confirmed in a more detailed fashion this progressive, worse and progressive nature of this disease at the level of various indices, including cognitive function, nigrostriatal degeneration, hyposmia. I'm not going into this in great detail, but we collaborated with Marios Polidis in this work. We actually sent a few of our subjects over to the UK in London, and they did functional imaging on the serotonin system. And what was very interesting that came up is that even some of the asymptomatic carriers seem to have a serotonergic deficit, even before there was any abnormality on the DAT scan. So that perhaps during the course of the disease, serotonin degeneration may predate dopaminergic degeneration, at least in this genetic form of PD. And we're generally very interested in the subject of asymptomatic carriers. So through the PPMI study, I mentioned the 8 asymptomatic carriers we had studied as part of the MEFOPA. Here we had a few more asymptomatic. We tried to do a more detailed phenotyping in these cases. We did not find much. That's the bottom line. That they were not that, here we compared them to real controls. And they were not that different, except that the MoCA was slightly lower, but more importantly, it seemed to correlate with a slight hyposmia that they had. So maybe there's something smoldering in terms of cognitive function and hyposmia, but it was not something that significant. That is why we've sought funding and actually got some funding from the Michael J. Fox to really look longitudinally at this cohort of asymptomatic carriers to really see over time, looking at various indices, both biological indices, clinical indices, imaging indices over time that reflect various areas of the body, even MIBG, which measures noradrenergic innervation of the heart, polysomnography to see if they have REM sleep behavior disorder. If these things develop over time and really following them longitudinally, and the aim there is to identify, it's a very grandiose idea, but identify where, when, and how does Parkinson's disease start? Because, they're not even at the prodromal stage, these subjects. They're before that. They are pre-prodromal, but almost predestined to develop the disease. As I mentioned, 80 to 90% penetrance. This is a study we did on polysomnography. It was not unexpected that almost all cases had REM sleep behavior disorder, but interestingly, not all. 9 out of 10 had some evidence of this. And on the asymptomatic side, essentially, we didn't find anything. And that's why we need to follow them prospectively to see which of these features, slight cognitive dysfunction, some biological index that alpha-synuclein is accumulating, some degeneration, either at the level of the heart, the noradrenergic terminals, at the level of DAT, which one of these develops first in the trajectory of the disease. So this is kind of a recap of what we've discussed. It's an accelerated form of PD, both in the onset and in the progression of the disease. especially regarding non-motor manifestations. There is this interesting phenotypic heterogeneity with extreme phenotypes, which we still don't understand how they come about. There is, again, but this needs this prospective study, but we have the impression that, the asymptomatic carriers remain asymptomatic, that seems to be happening, and then they start developing prodromal features quite rapidly, and they evolve into the motor phase of the disease quite rapidly. And that's why we need the prospective studies. So going on to biomarkers, we've done over the years various studies, and this is just a panorama of the studies we've performed, but I'm just going to focus on a few of these. One of the first we did, which may be interesting, especially to this audience, we looked at classical biomarkers of Alzheimer's disease. And this was in a small subgroup. And this was in collaboration with the lab of Betty Kapaki at the neurochemistry lab here at Genedis, and Giorgos Paraskevas. And what we found were these two interesting subgroups of two subject seats. The one on the left has very high total tau. These were exactly the two subjects I mentioned that had very early onset with a frontotemporal dementia-like presentation and very rapid disease progression. They both died, very, very soon in the course of the disease. And of course, they had this very robust, evidence by imaging also, neurodegeneration. So it's not surprising that they had high total tau levels. And there was this other small, again, subgroup of two subjects who had quite pronounced cognitive decline relatively early in the disease, and they had low beta amyloid. We've looked at various indices of alpha-synuclein biomarkers over time, but what has interested us from the beginning is actually red blood cells, which express quite a lot of alpha-synuclein. We've had studies in the past, but the more recent ones in collaboration with Evangelia Manolidou. I was just now in the PhD defense of Kostadina Demoula, and she just presented a thesis. Part of it was this work, and where she was able to show that in genetically undetermined Parkinson's disease, a specific ELISA for aggregated alpha-synuclein shows a higher signal in idiopathic PD compared to controls, whereas this was not the case in the two genetic cohorts, including the 83T PD. So in the erythrocytes, we don't seem to have, aggregated alpha-synuclein in this genetic subgroup. And it's quite complicated to discuss why that may be the case. Just here recapping a lot of studies, but perhaps it's not that, I don't think you will get much, just me going through them. Again, they're available for you to peruse, and whoever wants can, contact me if they need more information. But I think this study was very interesting. This is a study we performed, again, in collaboration with Leonidas Doxakis at the Biomedical Research Foundation of the Academy of Athens. And what Leonidas looked was really, can we have a window into the brain to see what's happening in the brain of these subjects? And a window into the brain could be microRNAs that are enriched, in neuronal microRNAs. So those are presumably coming from the brain. You're sampling the periphery, but they presumably reflect what's happening in the brain. And we had three groups, and the ones in bold show significant differences compared to controls. So there's a control group as well. And what is very interesting is that the two genetic subgroups, AFT3PD, AFT3PD and GBA-PD, behave very similarly in these assays across a variety of microRNAs, of neuronal enriched microRNAs, whereas the idiopathic PD had generally quite a different profile. So this, I think, speaks to the fact that these two forms have quite a few things in common, but so it's quite interesting. Another study we've completed recently, and I want to mention in more detail, is we looked at neurofilament levels. And as you all know, neurofilament levels, even the periphery, may reflect neurodegeneration. in the brain. And we just want to see whether our subjects, again, this is the PPMI cohort, were different from idiopathic PD and controls. And although there was a large scatter, they were different. A53-TPD had higher neurofilament levels compared to healthy controls, again, speaking to the fact that it is a more accelerated form of degeneration, whereas idiopathic PD were not different from controls. So this, again, summarizes this part with the biomarkers. It's a mixed picture. In some cases, our biomarkers go together with idiopathic PD. In some others, like the microRNA studies you saw, it was quite different from idiopathic PD. But I think this speaks to the fact that it's essentially the same disease, but the point of departure may be different. And of course, we would like to do more longitudinal studies in these biomarkers and to expand into omics. We have some collaborations, including a lipidomic collaboration, which has been published with some differences in lipid species in our group compared to controls. But I wanted to go into a very interesting genetic study that has been done. And this is an idea of Christos Koros from our clinical group. The question here was, okay, we know we have cognitive decline in our cohort. Is this influenced by ApoE4 status? And of course, it's known that ApoE4 may determine to some extent, correlates with cognitive function and progression of cognitive duration in idiopathic PD. And this is what we found also looking at our cohort of idiopathic PD. When we looked at the much smaller cohort of our APOE3T mutation carriers and comparing ApoE34 negative to ApoE34 positive, we found there was quite a significant difference in terms of MoCA score. So the ones carrying the E4 allele had a much worse cognitive score. This was not true in two GBAPD cohorts we looked at. There was no difference depending on the ApoE4 status. So this kind of triggered a series of thoughts, and including some data like the two subjects I mentioned that had much worse cognitive function, had lower beta amyloid, some indication, some functional imaging that some of our carriers may have A beta accumulation on PET scan, again, collaboration with Marius Politis. We had this idea that maybe if you have the alpha-synuclein mutation and you have the E4 allele, this could trigger the evolution of amyloid co-pathology. And this essentially is the idea that perhaps aberrant alpha-synuclein may secreted from neurons. It could be APOE3T, or it could be aggregated alpha-synuclein for other reason, impacts on astrocytes or microglia, could be the case, in an APO, and impacts on expression of apolipoprotein E4. And this triggers then release ApoE4, and this impacts on, again, back on neurons and instigates the amyloidogenic pathway in neurons. So this is an idea we have. Do we have any preliminary data along these lines? I think it's interesting we have data from iPSC-derived neurons. These are from controls or from carriers of the APOE3T mutation with who are Parkinson's disease patients. And what we see here is that if we apply conditioned medium from these neurons, iPSC-derived neurons, on astroglia, these are mouse astroglia in culture, we see an increased expression of ApoE. And this is also true if we use other forms of aberrant alpha-synuclein, such as PFFs, preformed fibrils, or brain-derived fibrils. We also get induction of ApoE expression in astrocytes. This at the RNA level, and we also have evidence that this also occurs at the protein level. So this is something we we want to study in more detail. This is a part of a grant we have now from the HFRI together with Era Taufik at the Pasteur Institute, where we combine these cultures of neuron and glial cells and aberrant alpha-synuclein as a stimulus to see if we can see this pathway indeed occurring in cell culture, but also we have experiments in experimental mouse models. So this wraps up this A53T part, going into the A30G. So this is another mutation which we quite surprisingly uncovered in the Greek population a few years ago. And this actually stemmed from a family which would seem quite straightforward that this would be an A53T family. We had many reasons for that. The subject, the proband, had an early age of onset of Parkinson's. He was very severely affected, cognitively especially, and psychiatrically, and he had a very clear autosomal dominant history of the disease. And he was from the Peloponnese. So no-brainer, right? He has the A53T mutation. Whereas not the case. So we collaborated with Tübingen, the lab of Thomas Gasser, and lo and behold, through whole exome sequencing, we identified a new mutation which had not been previously identified in the same gene. We thought there would be the A53T mutation, but actually it was a A30G mutation. We then went on to look at various cohorts from Greece and Germany, again stressing more familial cohorts, and we found a couple more families. They all had the same haplotype. They are shown here. And there was an indication perhaps of incomplete penetrance, as you see in these pedigrees. But that was substantiated in further work, as you can see. And we also, as part of the study, we did biophysical studies in collaboration with Markus Rextetter at from Germany as well. And Markus just used the recombinant A30G protein and compared it to the wild type. So what came out of this work regarding the structure, again, a recombinant form of the protein, is that there was a decreased alpha helical propensity. That's a normal propensity of alpha-synuclein. It seemed to disfavor binding to membranes, this conformation, the A30G, and it seemed to slightly accelerate the aggregation process of the protein. And this is similar to another mutation that's been recently identified, the V15A mutation, but it's quite different from most of the other SNCA mutations. Actually, in the last 4 or 5 years, although we thought, we've identified all possible, point mutations in the alpha-synuclein gene, a number of studies have uncovered new mutations, other new mutations in the SNCA gene that, segregate with Parkinson's disease in families in different ethnic backgrounds. And as I said, it's very interesting that this V15A mutation seems to have similar biophysical properties to A30G. And of course, the A30P mutation, that had been identified many years ago, also has a decreased propensity to bind to membranes. So we went on to do both genetic and cell biology studies studying this A30G mutation. We kind of widened our spectrum of investigation, including the majority of patients we have in this biobank. And what we found actually was 1.5% of patients in our biobank were positive for this mutation. The mean age of onset is 55. It has a lower penetrance. There's no specific geographic distribution. Only Greek patients identified. You know how Italians are very connected to us genetically. We've asked our Italian colleagues in their cohorts to have a very good kind of network there. They have not identified a single case, which is quite surprising. What's very interesting from this study, the second study, is that we've identified mutations even in sporadic cases. And again, this is because of incomplete penetrance. And this is kind of a grandiose statement, but if PD prevalence in Greece is 0.2%. Perhaps there may be 200 cases of Parkinson's in Greece with this mutation. So a large cohort where even perhaps intervention studies could be done. In any case, this is just more detail on the study I just mentioned. And I think the most interesting part of this study, okay, there are small numbers, but they're there. Sporadic cases with mid-late onset, right? So these are age of onset beyond the age of 50. They're completely sporadic, no family history at all, and yet 2 of them have the mutation. So you can identify them even in the wider population, even without a family history, without an early age of onset. And I sort of sell this as, alpha-synuclein mutations break out in the community. So going on, this is the last part of the study. I hope I'm not over time, but just to mention some studies we've done in, to understand the function of the protein more, the A30G SNCA protein in a cellular animal model context. What we've done, Marina Patozopoulos had a grant from HFRI, but also through our own support and through a grant from the GSRI, we've looked at cell models, including neuroblastoma cells, and also with Christina Kirousi in iPSC-derived neurons. Marina has also done some animal studies where she's injected wild-type and A30G SNCA PFFs in the well-known PFF striatal model of Parkinson's disease. And Konstantinos Padikaras has expressed the wild-type and the A30G SNCA in C. elegans. And he has some very interesting results. No time to go through all of this, of course, but just to mention the studies Marina has done, Marina Patozopoulos, in cell culture in the neuroblastoma cell line. And so we have 2 here. In neuroblastoma cells, we have cells expressing stable cell lines, expressing either wild type or the A30G protein, pretty similar levels of expression, but very different proteomic profile. You see here day 5 and day 7 for the wild type line on the right, and then day 5 and day 7 of differentiation of the A30G line. These cells are differentiated with retinoic acid and assume a more neuronal phenotype. And you see that they're very different between them. Some very interesting differences that arise on the biochemical level for alpha-synuclein, and both of them are shown here, and kind of reflect what Marcus Weckstetter had found in the isolated protein, the recombinant protein, is that the wild type goes to the membrane, whereas the A30G is very reduced in its membrane binding, as you can see here in the first line. This is an antibody against total alpha-synuclein. This is the membrane component for the wild type, and this is the membrane component for the A30G. All in the cytosol, they're quite similar, and this is reflected here. Very interestingly, phosphorylated alpha-synuclein is almost completely knocked down. So this A30G protein has a very hard time being phosphorylated. What's kind of even more interesting is that you have kind of spontaneous oligomerization, and if you will, aggregation of alpha-synuclein under these conditions of overexpression. And you see here the wild type, both in the soluble compartment and insoluble compartment, we see these higher molecular weight oligomers, and they're very much reduced in the A30G. So contrary what one would expect, A30G protein binds less to membranes, is much less phosphorylated as S129, and oligomerizes and aggregates, if you will, much less than the wild type form. Is it toxic? It leads to much lower levels of tyrosine hydroxylase, which is a classic kind of hallmark of what happens in Parkinson's disease compared to the wild type. So tyrosine hydroxylase levels are lower. Oxidative stress is increased, as you can see here. both day 5 and day 10 of differentiation. And there's toxicity. The A30G cells, over time of differentiation, although they start okay at day 3, over time of differentiation, they are lost in the cultures, and there's increased presence of cell death with this antibody against cleaved caspase 3. So a very dissociation, that a very interesting aspect of dissociation of toxicity from the classic aggregation, phosphorylation, and brain localization of alpha-synuclein. So what could be the target of this aberrant alpha-synuclein? One idea we have is that quite remarkable effects on the mitochondria so far. I mean, this is very preliminary, but we do see a reduction of mitochondrial volume with this mitotracker, which is quite specific to the A30G, as you see in the center, compared to the wild type and the control. And we have also, again, a similar, and this is the proteomic profile I showed you before, but this is just the mitochondrial matrix. And again, you see great differences in A30G line on the left compared to the wild type line on the right. And there's various mitochondrial proteins that are altered in these conditions. So maybe there's some effect on mitochondria, and this is one we want to study further. And of course, as I mentioned, we've generated with Christina Kirousi some iPSC-derived neurons from 2 of our subjects, and we're in the process of studying them. And this is not the study, unfortunately. This is the study from PNAS quite a few years ago, where fibroblasts from our A53T patients were used to generate iPSC-derived neurons. And this beautiful work by Rebecca Matza and colleagues showing that there is this axonal synaptic degeneration in this model, and some very preliminary findings we have from the present iPSC-derived A30G neurons seem to go in this direction, but it's still very early to say. So why should we care about genetic synucleinopathies? They are for sure synucleinopathies in the sense that we know synuclein is the primary cause, aberrant alpha-synuclein is the primary cause leading to neurodegeneration. So studying exactly how the disease evolves, biomarkers, a response to treatment, hopefully with clinical trials, may be very informative also for idiopathic PD. But again, we have to always be cognizant of differences and similarities that may reflect differences in the point of departure in this condition compared to Parkinson's. And I've been trying over the years to convince companies that they should invest in synuclein-targeted therapies in these cohorts, but hasn't happened yet. If you have any ideas, let me know. So again, at the more cell biological level, to understand the neurotoxicity conferred by these mutants, it doesn't seem to be homogeneous. Every mutation seems to be doing its own thing, so it's kind of puzzling. And of course, iPSC-derived not just neurons, but also glial cells. The group of Papastefanaki from Pasteur, again, had a beautiful recent publication on astrocytes, iPSC-derived astrocytes having a phenotype from the A53T cases. But also I'm interested in how these iPSC-derived lines may reflect the clinical heterogeneity we see in the cohorts. And of course, the asymptomatic carriers, again, not just how the disease evolves, but examining what perhaps is protective. For example, I'm always puzzled why that 87-year-old woman had never developed Parkinson's disease. So these are the contributors, main contributors for this work. Probably I've neglected some. I want to thank everyone, and including sources of support. Thank you.
Many, many thanks, Professor Stefanis, for this very interesting talk, shedding light on the pathogenesis of genetic cases of PD. I think it's time for questions. Please. Are there any questions?
Can I?
Yes, of course, Kostas.
Yeah, thank you. Wonderful talk. A pretty broad range of information. I just curious about 2 specific things. One of the pretty well-known co-pathologies involved in various forms of PD is TDP-43 co-pathology. So I'm curious whether that factors in some way to your mutant individuals. The second is, in the presence of co-pathology, one of the ways to assess the effect of the mutation specifically is to use CRISPR in iPS-derived whatever, whether they be organoids or assembloids or neurons. Any thoughts? Maybe someone is going in that direction using CRISPR to correct, and then comparing the CRISPR-corrected and uncorrected iPS-derived whatevers to try and understand specifically what the mutation is doing.
Yeah, thank you. Thank you, Costa. We go back a long time, so it's nice to see you again after so many years. Yeah, the TDP-43 pathology, we haven't looked into that very much. I'm not aware that it impacts a lot, Parkinson's phenotypes, or if there's any evidence for that. They may be out there, but I just don't know. There's a lot of talk about tau, but we're really not studying even that at this point. We're just studying the connection, possible connection, because I have to say, I want to present it here, but this is very, very, because of the interest of the group and because we got this grant recently to study it, but these are very, very preliminary results on the co-pathology and should be taken with a grain of salt. What we've shown already, as you saw, and that's goes to your second question, is that when we exposed astrocytes to aberrant forms of alpha-synuclein, there was increase in the expression of ApoE, and presumably that would also be ApoE4 if this substrate, genetic substrate, exists. And our idea is that maybe this accounts for the finding in our patient cohort, but this is still very preliminary. And of course, you're right, absolutely. We have to see if this effect is reversible with CRISPR-Cas, or in the project, what we've done is we've put, for example, is this effect reversed if we use a very specific antibody to alpha-synuclein? Will that reverse this effect on polyprotein E? And of course, CRISPR-Cas or other methods to have controls are needed. But we have, on the other hand, also the data with the PFFs and the other forms of alpha-synuclein that also induce this ApoE expression in astrocytes. So we're working kind of on all these fronts.
Great, many thanks. Are there further questions?
Can I ask
more
about the mechanism? I find quite interesting the work with mitochondria, because that connects with the synaptic role of synuclein and the protection based on different mutations, or aggravation of pathology based on different mutations. How would you think that will be connected, since synuclein is not, through the aggregation affects the mitochondria function? Or, I know it's a speculation, but what's the direction of the experiment you are developing, trying to understand how the synuclein mutation can affect mitochondria differently?
Yeah, we don't think it's aggregation, because at least classic aggregation that we can measure with these oligomers, oligomers are reduced both in the soluble and insoluble compartment. We haven't looked specifically at what happens in association of alpha-synuclein with the mitochondria, and this is part of what we want to do. We want to see if there's a different association of wild type versus A30G synuclein with the mitochondria using proteomic methods. We're collaborating with Maria Samyataki at Fleming for this work. I showed you already a wide range of mitochondria.
The reason I'm asking is because it could be,
but we don't know if it binds, for example, more to the mitochondria, or binds less to the mitochondria.
Because that could be another function for
bind to membranes. Means it binds to membranes much less than wild type. So it's puzzling to think it would bind more to mitochondrial membranes. But we haven't looked at that specifically yet.
Yeah, that could be very interesting because that could, give even a novel function for synuclein on mitochondria, other mitochondrial dynamics, or even the
Yeah, I
trafficking.
mean, we have to look also at things like, mitochondrial fission and fusion, things that are known.
Yes,
I mean,
yes.
others have studied, Haas has published on this, how alpha-synuclein may induce mitochondrial fission. Perhaps, A30G induces more mitochondrial fission or something.
Okay.
Yeah.
Thank you.
Further questions? I would like to pose a question. You presented data showing that psychotic symptoms are somehow more common in genetic cases compared to idiopathic PD.
Well, in the specific, we studied it quite at length, and that goes along usually with, the cognitive deterioration. It's the classic, profile of, I'm sorry, I'm getting dark here. Maybe I'll put the light on. Sorry, just a moment.
Okay, no problem.
Okay, so so it's sort of a correlate of cognitive deterioration, and they get this, visual hallucinations, delusions.
Independently of being on anti-Parkinson's medication?
That's a very good point. Usually they're at higher doses of anti-Parkinsonian medication, and we've shown that also quantitatively, compared to age and disease duration matched idiopathic PD patients. So that could account in part for the increased, psychiatric manifestations. Although over time, we increasingly try to avoid drugs such as dopamine agonists, for example, that would exacerbate these symptoms, and we tend to stick with just levodopa.
Okay. Thank you. Are there further questions?
Just the last one, if there's nobody else to ask.
Go ahead.
I know you're also checking from the plasma. Are you checking also the different profile of besides the microRNA that you did a very nice study with? You check the microRNAs of the different profile. Are you also checking, for example, exosomes or other plasma proteins that could give you a different function of brain status, health status between the three different categories?
Yeah, we've done. I mean, we haven't done specific studies in this cohort, and it was actually in one of the slides, something that we'd like to do. We've done some studies. I mean, it's not, well, it is a study in the sense that it has not been published. We've sent samples to Tony Wiscore from the plasma to do secretomics, and there's some interesting differences in our cohort, but that's just total plasma. But, exosomes is something we're putting in some grants here and there, and we hope to get funding to do that as well. Yeah.
Thank you.
Many thanks. Are there further questions?
Full opportunity.
Okay. So many thanks, Professor Stefanis, for this very interesting talk and for answering our questions.
Okay.
Take care. Many thanks. So see you during the seminar in April. Yeah. Many thanks.
Thank you.
Bye bye.
Costa.